Competition is, of course, everywhere in nature. With this being ABS, I'm sure I don't have to tell you that one widespread behavioral strategy for dealing with competition is that of overt aggression. But aggressive behavior can be costly, as it's very energetically expensive and can lead to injury or even death. And the competitive environments that demand aggression are seldom static. We can think of seasonal changes occurring in a relatively predictable way as animals move through different breeding stages or times of the year with more or less competition. But social environments can also change on faster timescales in less predictable ways, as individuals move around and encounter one another as potential mates or rivals. So it makes sense that animals should be able to adjust their aggressive phenotypes over both seasonal and social timescales to maximize the benefits and minimize the costs of aggression. And there should be some underlying physiological mechanisms that can facilitate any such changes in behavior over both timescales. One such potential mechanism that has been well studied in males is described in the challenge hypothesis. Originally shown in the male song sparrow, it has since been applied to multiple vertebrate taxa and generally states that circulating levels of the steroid hormone testosterone track both the seasonal and social environment. That is, circulating testosterone increases and decreases over the course of a breeding season so that males have a higher systemic T at times of greater competition and reproduction. But critically, males can also increase their testosterone levels rapidly within 10 to 30 minutes following a social stimulus, like getting in a fight. On both of these timescales, having higher testosterone can promote aggression and other competitive phenotypes, thus preparing or priming males for imminent social challenges. Now, of course, we know that females also frequently face intense competition, and females are capable of producing and binding androgens, albeit at lower concentrations than men. But we still know very little about how the challenge hypothesis applies to half of all vertebrates. So, in my dissertation, I have been systematically addressing questions about the role of testosterone in regulating female aggression over different timescales, using tree swallows as a study system. These songbirds are obligate secondary cavity nesters, which means they readily breed in artificial cavities or nest boxes that we put up. Since females must obtain a nest site to have any reproductive success in a given year, there is intense competition to get a box. And my advisor has previously shown that more aggressive females are more likely to obtain a nest when boxes are limited. Females must also frequently defend their territory from intruders who threaten to evict or even kill them for their box throughout the breeding season. And finally, testosterone is a good candidate mechanism for aggression in this system because we know that females are behaviorally sensitive to the hormone. When given testosterone implants, females become more aggressive but spend less time incubating their eggs. So, based on this experimental data suggesting that aggression is most useful during initial competition, but could interfere with parental care, I expect female aggressiveness to decrease over the breeding season. And if this seasonal shift in behavior is mediated by a seasonal change in circulating testosterone, then I would expect T levels to follow a similar pattern. I predict that females will respond to increased competition or the presence of an intruder with heightened aggression, and that this behavioral response will be followed by a rapid increase in testosterone production in line with the male challenge hypothesis. To address these research questions, I have measured aggressive responses in a lot of birds during my dissertation. And the main way I've done this is by using a classic technique called a simulated territorial intrusion. So in our system, this involves placing these taxidermic decoys directly on a nest box in this defensive pose for 5 minutes, and then quantifying the aggressive behaviors exhibited by the female box owner in response. So it looks something like this. And just as predicted, average female aggressiveness towards simulated intruders decreases over the course of the breeding season. I've also captured females and taken blood samples at multiple breeding stages to look at baseline circulating testosterone levels and found a similar pattern, with higher T levels seen early on and lower levels during these later parental stages. So far, this seems pretty consistent with the male challenge hypothesis. So, to address the social aspect of the challenge hypothesis, I again used simulated territorial intrusions, this time lasting for 30 minutes, after which time I caught females and took blood samples again to measure testosterone. And I found that these experimental females, right after a fight, had lower testosterone levels than date and stage match controls. And I want to point out that the timing of the sampling here is well within the typical time course during which testosterone is expected to be elevated in response to activation of the HPG axis. What's more, I found that the amount of time a female spent being aggressive during the simulated intrusion negatively correlated with her testosterone levels, suggesting to me that somehow just being involved in the aggressive interactions was using up testosterone or interfering with the production of more testosterone, or maybe both. To recap, in line with my predictions from the challenge hypothesis, I did see a seasonal decline in both female aggressiveness and their circulating testosterone levels. But rather than seeing an increase in testosterone levels after a fight, females appear to decrease testosterone levels right after a social challenge. I've been looking at subsequent changes in steroid processing in the brain and have co-advised an undergrad project looking at changes in the steroid production pathway in the ovaries that can partially explain these social results. And I'd be happy to discuss that more with anyone if you want to reach out. But from a behavioral ecology perspective, what I find myself really wanting to know is, do these changes in hormones and behavior over time affect survival and reproductive success? So, to look at one aspect of this, I've returned to this really rich data set I've accrued on seasonal changes in aggression. And I've been looking for relationships between various fitness components and female aggression at different breeding stages. And so far, two interesting patterns have emerged. First, early aggression is positively correlated with later chick growth and quality. So the most aggressive females early on have the best quality chicks. Second, this late aggression is inversely predictive of female survival to the next year. So females that are least aggressive during the chick period are more likely to be seen in the subsequent breeding season. So this suggests that over a lifetime, females that can go from high to low aggression over a breeding season should have greater overall fitness, but maybe not for the reasons I initially predicted. So to finish up, I think my results demonstrate that seasonal patterns of testosterone and aggression cannot be generalized to the timescale of more rapid changes in testosterone based on social interactions. And a growing body of literature from male songbirds actually suggests this pattern is not limited to females. It's my hope that this work can contribute to our understanding in both sexes of how hormones and behavior can affect one another to ultimately influence an animal's fitness outcomes. Thank you very much for your attention.